Good morning, students. Today we are going to study another protozoan, that is Trypanosoma cruzi. Trypanosoma cruzi is the causative agent for Chagas disease. This is also known as American trypanosomiasis. Now in this picture you can see the Redowik bug, that is the causative agent, and below it is a slide showing the peripheral smear. Here you are seeing the eyes. On the eyes, there is a swelling that is known as Romanas sign. Now, this is this trypanosoma is an hemoflagellate, and the classification of this is kingdom. It belongs to kingdom Protista. Phylum is Sarcomastigophora, subphylum Mastigophora, class Zoomastigophora, family Trypanosomidae, genera is Trypanosoma. These protozoans, that is the hemoflagellates, they require two hosts to complete the life cycle. Trypanosoma cruzi and Chagas disease. That is, as I told you, the etiological agent for Chagas disease is the intracellular protozoan parasite, Trypanosoma cruzi, which is transmitted by the vector. The vector is an insect named Redowik bug. Redowik bug bites the human host and transmits this parasite. Say that is what Trypanosoma that is formed when this Redowik bug is going to bite the human being. Trypanosoma cruzi, the organism characteristics is this parasite exists in two forms, that is a mastigote and trypomastigote. You can see this a mastigote form. It is oval shaped, 2 to 4 micrometers in diameter, and this is the multiplying form, and you will find this multiplying form in the Chagas disease. A mastigote form lives in the muscles of the heart, skeletal system, nerve cells, cells of the RE system, and so on. Trypomastigote is 20 micrometers long, and it is the non-multiplying form. This form you find in the peripheral bladder. Now the life cycle of this Trypanosoma cruzi, the definite host is human being, intermediate host is Redowik bug, and infective form is metacyclic trypomastigote. Now when this Redowik bug bites the human beings, the infected Redowik bug will transmit metacyclic trypomastigotes. This metacyclic trypomastigotes, they are deposited along with the feces at the site of the bite or at the site of the wound. From there, this infective forms, that is the metacyclic trypomastigotes, get transferred through contaminated fingers into the wound or the conjunctiva. From there, they will reach the RA system, spread throughout the body by spreading in the RA system, that is reticuloendothelial system, then gets converted into a mastigote forms. From metacyclic mastigote, tripomastigote forms, they will get converted into a mastigote forms in the RE system, RE cells, and starts multiplying there. During the multiplication, this tripomastigote will get converted to promastigote, epimastigote, and again tripomastigote form. And these tripomastigotes are released into the blood of the host. When a radioid bug bites the infected host, tripomastigotes get sucked in its blood meal. When these tripomastigotes are sucked as the blood meal, they are going to reach the salivary glands of the insect. From there, they get transformed into the epimastigotes and migrate to the hindgut, multiply and develop into metacyclic tripomastigotes. Radioid bugs bites the new host and infects with metacyclic tripomastigotes, and the life cycle is complete. So this is how the life cycle of radioid bug is causing Chagas disease. Now this infection not only transmits through the bite of the radioid bug, but it also can be transmitted through the blood transfusion and the placenta. When the pregnant woman is infected with this tripomastigotes, that is, with this disease, trypanosomiasis, the infection can be transmitted to the newborn. See, the Trypanosoman bug stay, that is, Trypanosoman bug takes the blood meal, passes as a metacyclic tripomastigotes in the feces, and the tripomastigotes is entering the wound, enters the bite of the wound or the mucosal membrane, such as conjunctiva. Then, once it enters the human being, metacyclic tripomastigotes penetrate the various cells at the bite wound site. Inside the cells, they get transformed into the mastigotes. These mastigotes multiply by binary fission in the cells of the infected tissue, and they are released as tripomastigotes. And these tripomastigotes can infect the other cells and transform into the intracellular mastigotes. A new infection sites develop, and the clinical manifestations can result from this infective cycle. The intracellular mastigotes transform into tripomastigotes, then burst out of the cell and enter the bloodstream. Once they are entering into the bloodstream, when the radioid bug takes the blood meal, then the tripomastigotes get ingested via the blood meal into the radioid bug. They will get converted into epimastigotes in the midgut, multiply in the midgut, and then get converted into metacyclic tripomastigotes in the hindgut, and after that they are going to reach they are going um, they will reach the new host, and that's how the life cycle gets completed. So trypanosomiasis or Chagas disease is caused by two hosts. That is, whenever the trypomastigotes they are going to complete their cycle in human being and ready with birth, then only the whole infection can be transmitted. Pathogenesis and clinical features. The mastigotes can kill the cells and cause inflammation consisting of mononuclear cells. Cardiac muscle is the most frequently and severely affected tissue. In addition, neurological damage leads to cardiac arrhythmias and loss of tone of the colon, that is megacolon, and esophagus, megaesophagus is caused. In acute phase of Chagas disease, the facial edema or a nodule is formed, that is known as chagoma, near the bite. This chagoma is coupled with fever, lymphadenopathy, and hepatosplenomegaly. This acute phase can resolve within 2 months, and most individuals then remain asymptomatic. But yet, few can progress into the chronic form with myocarditis and megacolon. Death from Chagas disease is usually due to cardiac arrhythmias and cardiac failure. During acute phase, there are both trypomastigotes in the blood and amastigotes in the intracellular tissues. In chronic phase, the organism persists in amastigote form only. So, pathogenesis you will see both as a acute disease as well as a chronic disease. In acute, usually you will find there is a chagoma or the inflammation at the site. Then there is fever, lymphadenopathy is also seen, and hepatomegaly can be seen. And the patient usually resolves in 1 or 2 months. In chronic, they will remain asymptomatic. That means fever, etc. is not seen, but it will slowly progress into the chronic form, and myocarditis and megacolon will develop, and this is the cause of the death of the person. Laboratory diagnosis. How will we diagnose this patient? That is by taking the peripheral blood examination. Now, what is peripheral blood examination? That is, you are going to take blood from the periphery of the person, that is from the skin. You just suck the blood and make the slide. Now, that is take a, like you do the HB, etc. Like that, you have to take a prick on the finger and a drop of blood, make the peripheral blood smear slide and stain it. Trypanosoma cruzi you will find in 3 forms, that is amastigote, epimastigote, and trypomastigote forms. Now, lab diagnosis. You're doing the blood examination. You can do by muscle biopsy. You can do the bone marrow aspirate, and Trypanosoma cruzi is seen even in the CSF. That is, yes, cerebrospinal fluid. When you stain with DIMSAs, you can see the blue colored Trypanosoma seen. The hypersensitivity test can be conducted, and you can do it with the Cruzi antigen. Treatment. The drug of choice is nifurtimox, benzimidazole. These are the two drugs that are usually used. Prevention, eradication of radioic bug nests, construction of homes lacking in cracks and crines, clearing the forest around the villages, using insecticides and helpful measures, as there is no vaccine available for Trypanosoma cruzi or the Chagas disease.
તમારે સાથે આવું પડશે લાગે તમારે સાથે આવું પડશે કે શું તમારે સાથે આવું પડશે લાગે કોને મને કેટલો આવશો હા તો આવશો કેટલા લાવીશ બાઈક પર થોડી આવશો કેમ નહીં આવે